Um, so now it's my tremendous pleasure to introduce our speaker for tonight, Paul Lewis. And I'm just incredibly fortunate to have known Paul for longer, I think, than either one of us cares to acknowledge. I first met Paul when his wife and my friend, Louise Lewis, and I were working together in a mycology lab, learning something at the time that was quite new called PCR. And it was during this period that I first met Paul at a lab party. And he was at that time doing a postdoc with Bruce Weir at NC State. So you know how it goes at a party. So I approached him and I said, "So what is it that you're working on, Paul?" And he said, "Well, I'm something like, I'm starting to mess around with Bayesian approaches to statistical phylogenetics." And so I think I just gave him a blank stare because at the time, I think it was the first time I had heard the word Bayesian, and this was in something like 1990. And I'm pretty sure that everybody here in the audience would agree that Paul was onto something important. So to give you a little bit of Paul's background, he received his PhD from Ohio State in plant biology, so he is a bona fide biologist, and he specifically studied the evolution of the polygonella. I'm not a botanist. He moved from there onto a postdoctoral position, first at NC State and then later at the Smithsonian, and in 1995 he began an assistant professorship at the University of New Mexico, but left for the University of Connecticut in 1999, where he has remained until this day. He is now a full professor in the Department of Ecology and Evolution with a joint appointment in the Department of Statistics. And over the course of his career, he has made so many contributions, it's really impossible to summarize, but just to touch on a few of the highlights, he has contributed to phylogenetic methods and theory, including, but certainly not limited to, likelihood analysis applied to morphological phylogenetics, something that has been terrifying to most of us, Bayesian phylogenetic inference, and innumerable computational tools for phylogenetic and population genetic analysis. The list goes on, but I don't want to cut into Paul's time. So with this, I will just thank Paul for speaking tonight and hand it over to Paul. Thank you, Paul.
All right. Excellent. Okay, it's hard to follow all of those acts that just happened, but let me tell you a little bit about what I've been working on, and it involves entropy and information in phylogenetics. So it kind of floors me how common words that we use all the time are so vaguely defined if you actually try to look them up. So if you go to the Oxford English Dictionary and ask, what is information? It says it's the imparting of knowledge in general. That's one of the main definitions, which just begs the question of what's knowledge and why can't it be specific? And if you look at different synonyms of information, in a thesaurus you get things like facts and data and then poop. But are these really synonyms of information? And what is information exactly anyway? So here's some data, 300 nucleotides from 25 sites and 12 taxa. But I suspect that nobody in this room would expect to use these data alone to reconstruct the history of those 12 taxa. There's a lot of data here, but not a lot of information. It's not to say there's not some information about some things. It tells us a lot about the rate of substitution, and it tells us something about the nucleotide composition. But in terms of information about the tree topology, if we're trying to infer the history of these taxa, we might be better off looking for some additional gene regions. So low substitution rates like this, I think of as like a photograph that's underexposed. So there's a lot of data there, a lot of pixels, but not a lot of information, not as much information as we would like. So here's the opposite extreme. In this case, I've simulated a really high rate of evolution by just drawing nucleotides at random from a discrete uniform distribution. And in this case, these high substitution rates are like an overexposed photo. So in this case, you still have a lot of data, a lot of pixels, but still not that much information. So the perfect substitution rate, the best substitution rate for inferring the history of a group of organisms is like a perfectly exposed photograph. The data that you have all the information you need to uniquely identify one tree topology out of all the possible tree topologies. Over the years, systematists and molecular evolutionists have worked out several ways of estimating information content. So I'm going to spend a minute or two going through those different, some of those different ways. I won't be able to get a chance to go through all of them, but go through some of the major ones. So in the 1980s, Walter Fitch did things like calculate the parsimony tree length of all possible trees and look at that for several different data sets. And what he found, the pattern he found was that for really noisy data sets that you would consider saturated with substitutions, these distributions, if you made a histogram, they were very symmetrical, whereas properly exposed and low substitution rate with the perfect amount of information, those data sets had a strongly skewed distribution, and there's this long left tail that leads out at the end to the most parsimonious tree. And that makes sense because it just means that the most parsimonious tree in this case fits the data way better than just any old tree topology that you draw out of a hat that contains all the possible tree topologies. And then for many years, skewness was a big thing, so you could use this G1 statistic to measure skewness of these tree length distributions. And so on the left there is a randomly chosen data that gives you a nice symmetrical distribution, and it's not surprising that the skewness statistic is close to zero. In fact, it's a little positive in this case, whereas on the other hand, if you have informative data that has a strong left skew to it, the G1 statistic is quite negative. Another way that people went about deciding how much information content was in systematic data was to shuffle or scramble the nucleotides within the columns of a data matrix, which basically erases the hierarchical structure due to the history, but preserves the character state distributions for each site. And then what you do is you calculate the tree score, the parsimony tree score, and you look where it sits amidst all of the shuffled data sets to see whether you can distinguish the original unshuffled from the shuffled null distribution. And a high p-value indicates that you have a really noisy data set, and a large proportion of the shuffled data sets have a parsimony tree score that's less than the one you observed. And then if you have perfectly exposed data, then you get this highly significant p-value. A third way that's been used is to focus on the entropy of composition of individual sites. So for saturated, highly noisy, overexposed data sets, you expect to see maybe all 4 nucleotides showing up at each site, and they might even be close to the same proportion. So you get these pie diagrams that are almost equally divided among the 4 nucleotides, which represents high entropy. And then you can distinguish that from better exposed, more slowly evolving data, because the nucleotide sites in those data sets tend to be dominated by one particular nucleotide, so you have very low entropy. And then a lot of folks over the years have built these plots that compare the p distance, or the proportion of differences between pairs of sequences, compared that to an estimate of the evolutionary distance based on a model, to look at sort of graphically how many substitutions have gone on in different data sets. And so if you compare second codon position data that tend to hug the equality line there with third position data sets, then which have sort of abundant evidence of lots of extra substitutions, then that's been used to argue that third positions are saturated with substitutions and therefore should be stripped out of data sets. And I would like to question that assumption and maybe say that sometimes that practice is even misguided. And I'll talk a little bit more about this later. In some cases, even in a case as extreme as this, which looks pretty extreme, the third position sites can actually have a lot of information, and maybe even more information than second position sites about the topology of the phylogenetic tree. So all of these methods, or none of these methods I should say, are particularly applicable in to Bayesian inference, and that's something that I've been involved in for many years, maybe not quite as long as Anne indicated, but I, I definitely have been interested in Bayesian inference for quite some time. And so I've been exploring how to measure information content in the Bayesian framework, which provides, interestingly enough, its own sort of built-in, very natural way to measure information content. I feel strangely compelled every time I give a talk to start with a coin flipping example, and so you guys are not going to be spared. So if we have a coin and we're interested in whether it's fair or biased in some way, we can flip it a few times to gather some data, but before we flip it, we come up with a prior distribution that reflects our beliefs before we start collecting data, and then that prior here says that we believe that it's just as likely that this coin that's sort of hidden in a box right now so we can't see it is a two-headed coin. That's just as likely as a two-tailed coin. Two-headed coin would have the model parameter here that the unknown in our model is theta. That would be 1 in that case. It always comes up heads. And that would be just as likely as a two-tailed coin that always comes up tails, or just as likely as anything in between, including a perfectly fair coin. We flip a coin only 3 times, and that already starts to transform our prior distribution, which is flat, into a more informative posterior that starts to allow us to consider some values of our unknown theta to be more plausible and some values to be less plausible than other values. And in fact, it already excludes the 2 extreme cases of a trick coin because we've already seen both heads and tails in our first 3 flips. Flip it a few more times, and that further serves to concentrate the posterior. So the flat prior has been transformed into a very informative posterior in this case. And so the key here is that information in the data is what takes that flat prior and transforms it or bends it into a more informative posterior. So the key to measuring information in the Bayesian world is to capture the difference between the prior and the posterior distribution. I have this little note at the bottom there to point out that Jeremy Brown in 2014 in a paper in Systematic Biology was, I think, the first person to start taking advantage of this approach in systematics. And so the stuff that I'm going to be talking about is sort of an elaboration on what he started in 2014. So my favorite definition of information is the one provided by Claude Shannon in his 1948 paper that gave birth to the entire field of information theory. So Shannon said that information is the resolution of uncertainty. Flipping our coin has eliminated some possibilities that a priori we were willing to entertain. And so the information in that data that we collected has resolved a lot of our uncertainty about whether the coin is fair or biased. But the uncertainty that Shannon was interested in resolving was which symbol was last transmitted over a telegraph system. So I want to spend a few minutes talking about how he developed this idea, and then we'll transfer to phylogenetics and try to transition to that. So if you have a sender that can choose one of 8 symbols and send it over the symbol somehow over a telegraph wire, then you have a receiver on the other end that has to somehow figure out what that symbol was that was sent. Shannon defined information as the number of questions the receiver has to ask in order to figure out which symbol was sent. So any one of these 8 symbols can be identified by answering 3 yes or no questions. And the sender can actually encode the answers to these questions in the form of binary digits that are sent across the wire. So for example, if the first question is it a circle or not, then the sender puts in a 1. If it's yes, if it's no, the sender puts in a 0 in the coding. The sender would encode a 1 if it's blue, a 0 if it's not blue, red. And then if it has a star, the sender puts a 1, and if it doesn't have a star, a 0. So, so you can see in this case that 3 binary digits sort of capture all the information needed to separate one of those 8 symbols from the other 7. So Shannon defined the term bit, so a bit of information is the amount of information that's represented by one of those binary digits, 0 or 1. So in this case, he would say you need 3 bits of information in order to figure out which symbol has come through. So as naturalists, we're all quite familiar with this definition of information. If you've ever used a dichotomous key to identify an unknown organism, then you know that it works by asking you a series of yes or no questions. Each of those yes or no questions represents a bit of information that you must have in order to get to the next step. And each step, each bit of information allows you to divide the number of possible answers by half. So it allows you to divide the uncertainty in half by answering each of these yes/no questions. The first question lets you cut the number from 4 to 2 in this case, and the second question, after you answer that, you've cut it from 2 to 1, and you've figured out what the plant is that you're trying to identify. There's a really strong relationship between entropy, and this is no coincidence because entropy was defined by Shannon to do this, to serve this purpose. There's a strong connection between the entropy of the distribution of symbols, that is how predictable the symbols are, that are coming at you, and how many questions on average you need to answer, or how many bits of information you need to answer in order to figure out those symbols. So if all the symbols are equally probable, the entropy is maximum, there's maximum uncertainty on your part about what could be coming next. If you didn't have any data, you would have no way of guessing which one was coming. So the entropy is 3 in this case, and that corresponds to the fact that you need 3 questions answered for every one of those symbols in order to figure out what it is. So what if some symbols are sent more often than others? So if, for example, one symbol, the blue starred circle, is sent half of the time, and then the other half of the time you might see one of 4 of these other symbols, then you might need to ask fewer questions because there's some predictability in this. You can, if that one symbol is coming at you every other time, then you can predict that symbol might show up even without any data coming into your receiver. So 1 bit is needed half the time, and the other half of the time you need 3 bits, as you can see there across the top. So on average, you only need 2 bits. You only need to answer 2 questions on average to figure out what symbol it is. And so there's no surprise that the entropy of that distribution is 2 as well. So if we take this to the extreme, if the sender always sends us the same symbol, then we don't need any information to figure out that what's coming is a blue starred circle. The entropy of the original distribution is 0, which means we need 0 bits of information. We don't have to ask any questions to figure that out. In the previous examples, there has been no noise in the system. There's no uncertainty at the receiving end. If we get those 3 binary digits, then we can just translate those and figure out exactly which symbol was sent to us. But if there's noise in the system, then that means that there's some erosion in the amount of information. So the 3 binary digits does not necessarily represent 3 bits of information anymore. And you can tell how much of erosion has occurred by looking at the entropy of the distribution at the end of the process, at the receiving end. So you can tell that there's some uncertainty about what symbol it was in. In this example, I'm presuming that the sender, there's like a 90% chance that the sender gets each of those digits correct, and a 10% chance that it's flipped from a 0 to a 1 or a 1 to a 0. So in that case, there's a 73% chance that you're going to get the symbol right each time. And so that entropy of that distribution at the end, it tells you how many bits of information that you've lost along the way. And so instead of 3 bits, this is more like 1.6 bits of information that is sort of provided by those 3 binary digits that we received. So another possibility is that the sender doesn't even send you all the information. So the sender only answers 2 of your 3 questions. Then there's going to be some uncertainty about, at the end, just like there would be if there were noise. So all we know, if we only have answers to the first 2 questions, is that we got a red circle, but we don't know whether it has a star or not. And so this uncertainty is manifested as entropy in that receiving end distribution, just like it would be if you had noise. And it's hard to tell the difference between noise and just lack of information. So what does all this have to do with phylogenetics? Well, in phylogenetics, our task is to identify one symbol out of a whole bunch of symbols, one tree topology out of all the possible tree topologies. And a priori, we have no idea which one could be coming at us for any particular data set. And so, we could encode these trees in terms of binary digits. So if you use 4 binary digits, that gives you 16 possible symbols that you could encode. We're only going to use 15 of those because there are only 15 trees, rooted trees in this case, for 4 taxa. So that leaves a little redundancy, which means that our 4 binary digits don't encode 4 bits of information. They only encode a little bit less than that, 3.9 bits of information. But the key here is that 4 digits is more than enough data to encode the 3.9 bits of information that we need to figure out which tree was sent to us. This is the point where I need to point out there's a big difference between data and information. So we could have used say, 100 binary digits to encode each tree. But since 4 binary digits is more than enough information to capture those 3.9 bits, the other 96 are just superfluous bits of data, binary digits of data. And so we don't really need that many in order to encode the information that we need. If these 4 binary digits were sent to us and there was no noise in the system, we could translate it, figure out which tree was sent to us, no problem, separate it from all the other 14. But binary digits is not how we get is not what our data looks like, is it, for phylogenetic trees? Instead, our data comes to us in the form of a data matrix that has a bunch of sequences or a bunch of morphological traits, and these traits or sequences evolved on the tree that we can imagine is being sent to us. And so the question is, how many bits of information are in those sequences that we got? How long do sequences have to be to capture 3.9 bits of information, which is what we need to figure it out? Well, we can get some intuition about this by picking one of these trees and simulating data from it, just as a little experiment. So obviously, if you simulate 0 bits of data, or zero sites of sequence, then you get no information whatsoever, and you have no basis of choosing. You might as well just choose randomly and uniformly from all 15. So the entropy of both the source distribution and the destination distribution in this case is the same. So the source distribution, we think of that as the prior distribution in a Bayesian context. The distribution at the end that tells you how much information you've lost is the posterior distribution. And in this case, with no data, those two are the same. The posterior will be identical to the prior, and they have the same entropy. As we add sites, the information grows, and we start to restrict the number of possibilities and being able to exclude some possibilities. And at this point, after 100 sites, under these simulation conditions, we have enough information to cut our uncertainty down to just these three trees, one of which is the true tree. So you can think of the prior distribution. The entropy of that distribution is telling us how much information we need. And you can think of the entropy of the posterior as telling how much information we're lacking. And so the posterior entropy sort of cuts into this pie, and what's left over is how much information is present in the data that we have to work with. So in this case, with 100 sites, we have 62% of the maximum possible information. If we go to 1,000 sites, we can nail that tree exactly. We have all of the 3.9 bits of information present in those 1,000 sites. And so there's no piece of the pie taken out by the posterior entropy because the entropy of the posterior is 0 in this case. And so 1,000 sites is enough to capture all the information. If we sequenced another 1,000 sites, we wouldn't increase the information anymore. It stays at 100%. It can't go above 100%. Can't go greater than 3.9 bits of information. And the reason for that is that any more sites that we add to this are just going to provide redundant information. It's kind of like if somebody comes to you and says, there's coffee ready in the meeting hall there, and then another person comes and says, oh, there's coffee ready in the meeting hall. You would say to the second person, thank you, but that knowledge has already been imparted to me. And so it didn't really tell us anything we didn't already know, and that's the same thing here. Another 1,000 sites isn't going to tell us anything we already don't know. So you can do some simple simulations that just show that this way of measuring information, by measuring the difference between the prior and the posterior entropy, works sort of as you would expect. So we have, for example, if the relative rate 1 is sort of perfect and that it gives you 100% information, the substitution rate is just right, then if you decrease that drastically down to 0.01, or you increase it up to 100 times that ideal rate, then you're going to lose information in both of those directions. If you add missing data, you're also going to lose information. If you have 100% missing data, you're not going to have any information, obviously. And as rate heterogeneity, site-to-site rate heterogeneity increases, you lose information as well. And that's because if you have high rate heterogeneity, that means that a lot of sites are evolving very slowly, they're underexposed, and a few sites are evolving very quickly, they're saturated or overexposed, and you don't have that many sites that are evolving at the perfect rate. So information is about resolving uncertainty, but just because you've eliminated a lot of uncertainty doesn't mean that what's left is the truth. You can, if you question a bunch of data sets and they all lie to you, then there's no way to figure out what the truth is. But one thing that we can do is query a bunch of data sets and we could see if their stories match up at least. We can see if they're singing the same song. So if you imagine each data set singing its own tune or song, you can ask whether those songs are consonant or dissonant. And so I want to talk a little bit about how we can use information theory to help us decide whether two data sets agree or conflict in the information they're providing. So David and Wayne Madison have both told me at different times and in no uncertain terms that I should put a picture of an organism in every once in a while into my talks. So behold sanguinaria, which is bloodroot. I took this picture on Sleeping Giant State Park in Connecticut a few years ago. So this is not entirely gratuitous, this image of an organism, because sanguinaria provides a really interesting case of horizontal transfer. The RPS11 mitochondrial gene is split in half, and the 5' half has undergone sort of normal vertical inheritance. And if you reconstruct the tree from that, you get sanguinaria placed with its relatives in the family Papaveraceae, the poppy family, just like it should be. If you look at the 3' end, it clearly has been horizontally transferred from some monocot. So the tree is quite different. So you can imagine these two data sets are singing very dissonant songs. They're conflicting with each other quite a bit. So it's a nice case where we know that there's conflict, and we can ask what happens if you measure information content in these separately and together. If you look at the 5' data alone, it turns out that there's about 75% information. So it captures about 2.9 of the 3.9 bits of information that we would need in order to get this tree exactly right. The 3' data also capture a significant amount of information, 67% or so. And so what would happen if you put these together and analyzed the entire gene? What kind of information content should it have? So if we concatenate these two data sets, put them together, analyze that with a Mr. Bayes or some other Bayesian program, you get a sample of trees from the posterior. That's what we use to measure information content. We're looking at we know what the entropy of the prior is, and the sample of trees lets us calculate the entropy of the posterior, and the difference between those tells us how much information we have. So what do you expect would happen if we looked at this? Well, I don't know what you think would happen, but what I thought would happen is that this conflict that we know exists in these two data sets some of the information in the second data set would cancel out the information in the first data set, and they would give me a posterior that had more entropy, more uncertainty than either data set alone. And so the information would be higher for individual data sets and then actually go down when you added them together. But that's not what you see. What actually happens is that when you concatenate these, you get 100% information. The posterior has 0 entropy. But the interesting thing is the tree that it identifies, one tree out of all the 15 possible that it identifies is neither the 5' tree nor the 3' tree. So what's going on here? So information is all about excluding possibilities. And what happens is each of the halves of this gene exclude the tree that the other half likes. So for example, the 5' data says there's no way it could be either of these two trees. One of those two trees is the 3' tree. The 3' data, on the other hand, says there's no way it could be either of these trees. And one of those trees is the 5' tree. And so what you're left with is this tree that's mediocre according to every site. And it's the only tree topology that shows up in the posterior sample, and so it looks like there's 100% information. But what you've got is a really terrible, it's like the worst tree for both sides of the gene. What's particularly nefarious about this is that if you look at these numbers at the bottom, it looks like everything's just rosy. You have 75% from the first gene, you add some information from the second half of the gene, it looks like it's adding information the first half didn't provide, and so you end up with 100%. So how are you going to know that these two halves of the gene are having this vicious argument by looking at just those numbers? Well, we've come up with a way that we call information dissonance to measure the dissonance in the songs that these different data sets are singing. And the way you do this is you generate a tree file, a posterior sample from each data set independently, and from each of those tree samples we can estimate the information content, and then we merge those into what we call a merged tree sample. So we basically just pool all the trees from both tree files into one tree file and analyze that as if it had been produced by one analysis. And we look at the difference between the average information content of these individual tree files compared to the information content of this merged one. What you would see if you looked at two data sets that actually don't have any that are actually consonant. So what if you took the two, for example, the two runs that Mr. Vase does that are independent but on the same data? They should say the same thing. They shouldn't be disagreeing with each other if they've converged and everything. And so this is what you would expect if you saw that. You would see the same information content in all three cases. So this would be an example of no dissonance whatsoever. But in our case, we're comparing 5' and 3', and you can see that the information content in the individual data files is much higher than it is in the merged. And so if you average these two guys, compare it to this guy, the difference between those is dissonance. And we're finding that that's a really good way of measuring how much conflict there is between two different data subsets. So it would be really nice if we could take the total information content of a data set and divide it up amongst clades in an additive way so that you could sort of see where in the tree the information is most abundant. And it would also be nice if you could do the same thing for dissonance. So if you want to see where's the conflict in these two data sets, is it just one clade or is it scattered all over the tree? And so let me talk about additivity. And to get this additivity, what we do is we break apart the trees that are in our sample and take apart their clades and build up a Larget-style conditional clade graph. So I'm just going to go through and take the clades from these two trees and build up a conditional clade graph. And these probabilities here show you the probabilities that each of these splits have, given that that is the parent. The nice thing about this conditional clade graph is that you can use it to kind of extend your sample. It's like you can add water to your coffee and make more coffee, but hopefully it's still good coffee at the end. So what you can do is mix and match clades in the conditional clade graph, and you can accommodate your sample for trees that you didn't actually sample, but which you probably should have sampled if you had sampled a lot more. So if you'd kept your MCMC going a lot longer, you probably would have seen these combinations. And so instead of two trees, we've doubled the size of our sample effectively by using this conditional clade approach. And once you have the conditional clade graph, it turns out that you can calculate the entropy and the information and dissonance and all these things for each clade, and then they all add up to the total. So in this case, there's 0.6 bits in each of those clades, and then a whopping 6.7 in this basal clade. And so it makes sense that the most information in this case is in that basal clade, because that split is present in every one of our trees, and it also it takes the 945 possible trees and reduces it to 9. That split alone does that. The rest of the tree just takes the 9 and reduces it to 4. And so the majority of the uncertainty is taken care of by that basal clade. So we can take apart the total information and show it by clade, which is kind of cool. You can do the same thing for dissonance as well. So here I simulated two data sets from two model trees, and the two model trees that I used differed only by swapping two of the leaves. And so if you do that, the merged consensus tree at the end of the day collapses that clade, and all the dissonance is concentrated in that one clade where the problem occurred. So while I've been talking, there's been this huge storm, these storm clouds that have been brewing above us that you didn't notice. And there's, so I need to talk about this storm, this dark storm that's about to happen. But then after that, I hope that we'll see a rainbow at the end, and I'll tell you a little bit about the rainbow at the end. So what is the storm about? The storm is about the size of tree space and scaling all this up to sort of reasonable sized problems. So there are 5.6 times 10 to the 26 distinct tree topologies for only 24 taxa. It's not a lot of taxa, as you know. We often look at problems way bigger than that. But thinking about this number very seriously, the 5.6 times 10 to the 26 can really make you weak in the knees. So you've all seen these kind of comparisons before, but one of my favorite is if you had a computer that could look at 1 billion trees per second, then you would have to start at 17.9 billion years ago in order to look through all of those trees. Another way of thinking about this that's maybe more relevant to what I'm about to say is that if you had an MC sample of 1 trillion trees, some of you might be approaching that now, but if you had an MCMC sample of 1 trillion trees, that still would be 564 trillion times too small to sample each of those trees once. So what that means is that we're just deluding ourselves if we think that we can estimate the entropy of the prior distribution in this example. And the prior distribution, remember, if there's zero information, that's the same as the posterior. And we have to estimate those two. The prior we can figure out analytically, but the posterior we have to estimate. And so the problem is that it looks like there's a lot more information than there really is. So if we take a sample of 10,000 trees for various cases of different numbers of taxa, we do quite well at estimating the zero information content until we get to this point where the number of trees starts to outstrip the number of samples that we have. So we have more trees possible than we have samples, so we're leaving some of them unsampled. And we get down to this case, what this looks like to information theory is that we've taken 654.7 million trees that were possible, and we reduced that to just 10,000. And so it looks like a huge amount of information. We've taken all of this uncertainty and reduced it to just that little dot up there. That looks like a lot of information, but really all that means is that this is how much tree space we've been able to cover in the time that we've been doing this analysis. So there's zero information, but this tells us there's 55% information. So that's a really bad thing. And this is only 12 taxa. So this made me almost give up on this project a couple of times, but there's sort of a bright spot here, and it involves a prior that is not present in a lot of phylogeny software. So we'll try to get this implemented in some form or fashion so that people can use this at some point. But the polytomy priors make it possible to visit, for an MCMC analysis, to visit trees that are less than fully resolved. For example, an MCMC analysis could visit this star tree, which only has one internal node. And it could also visit trees that are fully resolved that have, in this case, 4 internal nodes. And this resolution class is defined to be the number of internal nodes. So if a tree is in resolution class 4, it has 4 internal nodes. And what's kind of odd about this and counterintuitive is that using this prior will help us estimate information content despite the fact that it more than doubles the size of tree space. You would think that we already have this gigantic tree space that's causing the problem. Doubling it shouldn't help. But let me show you maybe how it does help. So what we can do is we can set up our polytomy prior so that all of these resolution classes are visited. If there's no information, they're visited about equal amounts of time. What this effectively means is that the star tree up here basically has a prior probability that's about 105 times larger than any of the individual tree in this most resolved class. And that star tree would have a prior that's 25 times higher than any tree in this resolution class 2, for example, because there are 25 trees in that class. But if you run without data, run an MCMC analysis without data, or run an MCMC analysis on a data set that has no information, then you should see equal values for each of these bins. So the good news is that you do see this. So even for this 24 taxon problem, in which the age of the universe is not long enough to estimate the entropy very well for the standard prior, if you use this resolution class prior, we can sort of think of the bins instead of the individual trees as our distribution, sort of this marginal distribution, and we can estimate this has an information content of 0.026%, when in actuality it has 0%. So that's not much error. And we've only had to sample 10,000 trees to do this. So you can see that we're supposed to sample each of these about equally, and a sample of 10,000 does a pretty good job of showing us that. So if there's no information, we can detect it, no matter what the size of the problem is. And then what's nice about this is it doesn't really hurt us when it comes to data sets that have lots of information. So in this case, I simulated data that was perfectly exposed, that had the right substitution rate, and all of the posterior is concentrated not only in this most resolved class down here, but it also is all on just one of those 5.6 times 10 to the 26 trees that are in that class. So it doesn't seem to hurt us to use this polytomy prior even when we have lots of information. Let me return to this example, which I was talking about how people often use these kind of plots to show that third positions are saturated and you should strip them out of your alignments. If we measure information content for this particular data sets, we see that third positions actually have more information than second positions about the tree topology. And the clades that are present in both of these trees are comparable, so there's not conflicting information going on here. So third position sites just are better exposed. They have enough information to figure out more clades than the second position sites. Now, because this example has 33 taxa in it, which is a lot bigger than 24, you might be suspicious after what I just said about the storm, scaling storm. So let's look at the polytomy prior in this case. If we use the polytomy prior and look at these distributions across the resolution classes, we see that most of the trees in our sample are in the most resolved resolution class for the case of third position sites, whereas most of the trees are in this resolution class that has 13 fewer nodes than a perfectly resolved tree for the second position sites. And if you measure information content, it doesn't qualitatively change. There's still way more information in the third position sites than the second position sites. So I think that in this case, it's safe to say that third position sites actually have more information than second position sites. And so you shouldn't throw out third position sites just based on a plot like this, because that doesn't tell you the whole story. This plot is based on the worst possible taxon sampling. Every one of those points is based on just 2 sequences, whereas this is based on all the sequences together. And so that gives you a lot more control over your estimation. So I just want to finish by going through very briefly a few reasons why I think it's important for us to measure information content. First of all, we might be interested in whether morphology or molecules has more information, and we might be interested in whether they disagree and where they disagree on the tree, for example. So if we can partition dissonance or partition information, we can ask where is the information and where is the conflict between these morphology and molecules. So that's something that a lot of people are keenly interested in doing. We can, as I just mentioned, we can use information content estimation to inform our site stripping. So we can take out sites that actually don't have a lot of information because they're way too noisy, and we have a better idea of which ones those are if we use information content measurement. Genomic data is sort of rife with various kinds of missing data, different patterns. So we could use information content to see which of these is actually costing us the most information. Is it better to miss taxa or miss genes or just miss data randomly? We can partition conflict among genes and ask where is the dissonance in the tree. If the dissonance is concentrated around a tiny internal node, then maybe we would suspect incomplete lineage sorting. So it gives us some tools to ask sort of what process is responsible for dissonance. And then information profiling phylogenetic information profiling is currently done by measuring the average substitution rate and asking prospectively which time periods new data is most likely to inform. Information content estimation provides us a way to retrospectively ask what time periods were most informed by the data we already have in hand. So it provides a different perspective. And finally, divergence time analyses. This is something we haven't ventured into yet because this doesn't involve discrete tree topologies. It involves continuous parameters, and entropy measurements on those are a little more tricky. But there's definitely a reason to look at this because in divergence time analyses, the prior is vitally important in helping us separate rates from times. And the prior carries with it a lot of information itself. If you calibrate a prior, then you're basically adding information to the prior. And so it might be hard to tell whether the credible intervals you're getting are due to the prior or due to the information in your data unless you measure the information content in your data. So I'll stop there and just want to thank NSF for supporting this work, and the Department of Ecology and Evolutionary Biology at UConn, which has given me a job for the last 18 years, and it's a really great place to work. I love my colleagues there, and my UConn collaborators, some of them are in the statistics department and some are in the Ecology and Evolutionary Biology department, and we've had a really good working relationship for the last 10 years or so. And I'll point out that this paper that talks about a lot of this is in Advanced Access in Systematic Biology, and I would especially like to thank the reviewers and the editors and associate editor who gave me lots of great feedback and prevented me from saying things that I would have really regretted later. So then also the slides that you've seen here are on SlideShare, so if you search for Paul Lewis on SlideShare, it's the only thing I've uploaded so far to SlideShare, so you should be able to find it in case you want to look at those slides again. So happy to take questions. Thank you. If you can go to the microphone, I can hear better. If anybody has any questions. Yep.
Great talk. I had a quick question. So the information that you compute, that's assuming the model has generated the data, right? So first of all, is that a fair understanding? And if it is, how much do we need to worry about that?
Well, the answer to all of those kind of questions is we need to worry a lot. Yeah, that's true. We're assuming that the model is not misleading us, so that would be another source of false information. And we haven't looked into model violation and information content at all, so I have no idea what to expect if the model's wrong. Good question. All right. I'll be around for the rest of the meeting, so if anybody has any other questions or wants to talk to me about this, I'd be happy to answer other questions later. Thanks.